Okay, as I said, I'll be brief. First I want to go over sort of how to do the lab, but as I'm talking about that, I would like you to be writing down what are the dependent variables and what are the independent variables for this lab. Okay? So what is a dependent variable? The thing you measure, right? The response, the thing you measure. Very good. What's the independent variable? What you're manipulating, right? What is the thing that we are testing? Okay? And there's going to be multiple for this experiment. Okay? Cool. So we'll do that after we go through. All right. Welcome to the Dark Adaptation Lab. You guys know a little something about dark adaptation, right? What is dark adaptation? Somebody tell me about it. Yes, Justin.
It's basically when your rods manipulate their photopigments and some other.
Yeah, so it's when your rods are fully bleached in bright light, so they have no photopigment available. And as you sit in the dark, photopigment starts building up, and so you're able to see better and better and better in the dark until you reach all the photopigment that you can have in your rods, which takes 15 minutes, 20 minutes. I'm going to give you 5 minutes, 15 minutes. And then you can see fully, right? Okay? What else do we know about dark adaptation? Does it happen the same threshold for all colors? No? So which colors, tell me about it. Aaron, you want to try? It's okay.
Blue and green are more intense, so you see them more in the dark.
They're more intense, but there's one photopigment, rhodopsin, for the rods, and it is most sensitive to green light. So therefore, green light can activate it the best. It's less sensitive to blue light. So in terms of threshold, you would predict what? What would green have? The rods are most sensitive to green. What would you think about the threshold? A lower threshold, thank you, Grace, compared to blue. What about red? Doesn't pick up red, right? So what's the threshold for red going to be? Really high. It's going to be cones, right? So whenever your cones quit working, whenever the light's too dim to activate your cones, you're not going to be able to see red. Does that make sense? That's what we're going to measure in this lab. All right? We'll do some psychophysics. What's psychophysics? That was day 1 stuff. 1 week ago, right now. Yes. So it's taking a physical stimulus and measuring the behavior, not the neural signaling, right? We're not sticking you in an MRI machine or anything, right? fMRI machine. But it's measuring your behavioral response. And all of our labs are going to be psychophysics, because you don't want me putting electrodes and stuff in your heads, right? Okay? So we're going to give physical stimuli, and you're going to give responses. That's psychophysics. Okay? Cool. All right, room 229D. These rooms are located in what we call the cognitive suite, or cognitive testing rooms, which if you go in the psych suite, it's that door towards the back corner, that corridor. And when you go in there, these are the 2 rooms that are on the right-hand side. They're labeled D and E. D is the testing room. It has a round table set up like this. Okay? At that round table is an old computer. Why do we have such an old computer, Dr. Pittman? Because these LED screens don't get dark enough to allow us to light adapt. They emit enough light that it prevents us from dark adapting. These old two screens actually are dark enough they don't emit light, and we can darken down with them in there, right? There will come a day when that breaks. I don't know what I will do. I have one backup in the lab. I'll probably retire, but I don't know. That'll be someone else's problem. Okay. There are two PowerPoint files located on the center of that screen. One is called Dark 1, one is called Dark 2, right? You're working in pairs. Does everybody have a partner? Who is Ariana's partner? Since she's not here. Okay. And you guys are going to do a 3. Sure. Okay. So, one of you will be the subject in this room, sitting in this computer, right? One of you will be experimenter in the room next door, sitting at this computer, right? And on this computer, I think IT is actually going to replace this computer. Actually, they can't because we're in the middle of doing this. So I'm going to have them not replace this computer, so we'll be fine. But on this computer, there are 2 Excel sheets. Guess what they're labeled? Dark 1 and Dark 2, okay? So if you're the experimenter, you're going to see all the words that the subject's trying to read, and that will bias you for when you're the subject. So have the first person do Dark 1, right? And the second person do Dark 2. If you're an experimenter, don't open the Dark 2 and look at it so you don't see the words that you're going to be reading. Cool? My trio, y'all got to figure that out, right? So maybe one person's not there for the first thing or something like that. I don't have a Dark 3. Okay. Yes?
I don't have a partner, so if it would be easier to split up the trio.
You have a partner now. Okay? We're all good? Everybody's got a partner? Cool, cool, cool. Okay, going back to this room. All right, so I'll talk about the stimuli in a minute. But one more thing I want to mention, there's a red floodlight on the floor, okay? I want half of you to be doing this lab in pure darkness. So when you shut that door, there's no windows in that room. It will be purely dark. We've taken all the other light sources out. Don't have your cell phone out, because you won't be able to fully dark it out, okay? And the other half of you, I want to leave this red light on while you do this, while you do the test, okay? So what would you predict about that? It's pretty light in there with that red light. Yes? It's not going to play a role because rods are not sensitive to red light. If rods, in fact, are not sensitive to red light, we should be able to prove that there shouldn't be an effect on our ability to darken light. Right? Does that make sense? What does that sound like? Okay? All right. Okay. When you go in that room, there are 2 floodlights and this other LED light in there that are aimed at your head. We want to make sure that you are fully bleached, that you're starting at 0 rhodopsin when you start each dark adaptation period, right? I used to make students sit there for 3 minutes. I'm feeling nice this year. You're only got to sit there 1 minute, okay? I think 1 minute's plenty of time to bleach all the rhodopsin. No reason to make you sit there 2 more minutes, okay? Now, if your data comes out all whack, then I'll be like, uh-oh, we should have done 3 minutes. But that's not an IV, okay? All right. Right here, so all those lights should be turned on with these little light switches here. So all you have to do is turn this power supply, this surge protector. That's the on-off for all 3, okay? When you get in there, you're going to open up, if you're the first one, you're going to open up Dark 1. Oh, before you start, in order to determine, we have to randomize who gets the red light. I don't want everybody in Dark 1 to all do red light. That would be a confound, right, with our stimulus sets. So, flip a coin, guess the number, I don't care. Some play rock, paper, scissors. Somebody figure out a way to randomize who's the red light and who's not. But don't everybody do the first person's red light and the second person's not. Randomize. Cool? Cool, cool. All right, so you're going to come in, open up Dark 1 if that's your first person. turn the light on or off, depending on what your thing is. Shut the door, turn the lights off in the room. Floodlights are on, right? You're going to light it up. Okay? All right. Questions so far? Okay. In the stimulus collection room, the experimenter room, there are 2 walkie-talkies. Kate, will you give me the walkie-talkies? Thank you. There are 2 walkie-talkies that I will probably demonstrate how to use. They are plugged into a USB cord to keep them fully charged. Be nice to your classmates. Plug them back in when you're done so someone doesn't show up in the lab and the walkie-talkies are dead. And then they're like, oh, we can't do lab like we planned to. Okay? So they should be plugged in when you get there, plug them in when you're done. Each person needs a walkie-talkie. You have to be able to talk to each other between these 2 rooms. And again, we can't use cell phones because we don't want light. Okay? Cool? All right. There's a timer. Now, if you want to use the timer on your phone, I don't care because you're in the light here, right? But there is a timer there you can use. You can use the timer on your phone. And there's also a minute conversion sheet because when you're collecting the data, we want it in seconds, right? So on your timer, when they get to word number 5, maybe it's at 4 minutes and 35 seconds. You need to be able to look at this sheet and go, 4 minutes is 300. Is that right? Yeah, 300 seconds plus 35 seconds. So you just unplug them, right? Leave the plug there. And then, where's the one?
On the bottom left.
This one. Okay, so there's an on button, all right? And so the experimenter's going to say, or the subject's going to say, okay, I am light adapting for 1 minute. You're going to start your timer for 1 minute and make sure they do it for 1 minute. When it gets to a minute, what are you going to say, Kate?
What?
It's okay.
If I can see it or not?
No, I'm the subject. You're the
Okay.
experimenter. I'm telling you I'm light adapting for a minute. You're timing me. When it gets to a minute, what are you going to tell me? Turn off the lights. Turn off the lights and start the experiment. Okay? Tell me.
Turn off the lights and start the
Push the button down.
Turn off the lights and start the experiment.
Okay. That's weird. These are kind of funny. But you have to hold down and talk, and then you have to let go so the other person can talk. Okay? If you're holding down the whole time, they can only hear you. Okay? So she's going to say, okay, you can start it's been a minute, you can start the experiment. Okay? All right. Then I'm going to say, okay, I'm turning off the floodlights now. And when you turn off the floodlights, you're going to go to the next slide on the PowerPoint. She's going to start her timer, and then you're going to read the words that are on the screen. Okay? 3 sets of words. First set of words is in blue. Second set of words is in green. Third set of words is in red. We're going to light it up in between each one, so we start out fully bleached for each set. Thank you. Questions so far? Good? Okay. When you open up the PowerPoint, this is what it's going to look like, right? Well, the instructions are right there. There's also these handy-dandy instructions that are on the Moodle site. There's also a copy already printed out for the experimenter to see. Okay, light it up. This says for 3 minutes. I've changed that. I didn't change it on this example, but I've changed that in there, 1 minute. Okay? You're done with your minute. Your experiment says, okay, you go to the next slide. You are going to say, okay, I'm turning off the lights, going to the, or you go to this and say, I'm turning off the lights, going to the next slide. Boom. And then you're going to read whatever word, it won't be word, but there will be a 4-letter word in the middle of the screen, and you're going to sit there and wait for rhodopsin to build up until you can read that word. And then you're going to say, you're going to press the walkie-talkie saying, word, and your experimenter is going to say, correct, and write down the time. Or, nope, try again. You thought it was word, but it's really long. Don't tell them what it is. But if they have it incorrect, say no. And you have to sit there and try until you get it. When you get it, when they say correct, you just hit the screen and go to the next one, which is less bright. Okay, and then you're going to wait until you can read that and say less. We're going to tell you if you're correct or not, write down the time, and so on. And it will get dimmer until there will be a point at which, you guys aren't dark adapted, Aaron's like, you can't see. You will be, rhodopsin will be building up in your rods. But there will be a point at which you get to that it will be so dim you can't read it. Once you get to 15 minutes, the experiment will say, okay, you're done, that's 15 minutes, you're fully dark adapted, you can't see it, you're not ever going to see it, okay? Then you can just hit forward, hit enter, till you get to here. This is when you have to turn the floodlights back on, which will be painful for a second, because you're bleaching all the rhodopsin that's built up, okay? So you're going to turn them back on for a minute, okay? After the minute, you're now going to do green, same thing, okay? Cycle down through green, okay? And what are we most sensitive to? Green, right? So green goes pretty fast. So like when it's probably going to be too fast for you to type the numbers in. You might want to have a piece of paper as the experiment to write down what the times are, and then you can type them in when it slows down. Okay, because you want to be like, correct, next, correct, next. Light adapt, red, green after 15 minutes, tell them to go forward. Okay, red, same thing, okay? All right, 15 minutes, now you're done, okay? Cool, please don't leave the spotlights on in there. They're actually LED. I used to have like things that got hot. I was always worried about fire. These are don't get hot because they're LED. But just don't leave the spotlights on. Cool, okay. This is an example of what the stimulus sheet looks like. You're going to put their initials, the subject's initials, 2026, whether it was dark 1 or dark 2, that's stimulus set. They're doing dark 1 or dark 2. But you're going to be, everybody in this one should be dark 1 because you're going to open the Excel file for dark 1 or dark 2 because the words are different, okay? You're going to put if they're 0 for no light, 1 if they're in the red light, and then over here, you're going to put their time. So here's an example. It took the person 12 seconds to read that, 17 seconds, 26 seconds. We're doing cumulative. You just let the timer go for 15 minutes. This person got all the way down to this point, and then they couldn't read these 2 last words, right? So you told them after 15 minutes to go on to green. Afterwards, you need to put 2 things over here, right? So this is the intensity going down. So what intensity did this person get to? 3, right? That goes here, and that lowest number, or the biggest number goes there. So for this one, you would put 3. Oops. I'm not going. 3 and 277 was the time it took to get to 3. Cool. Then you go to the green tab. Same thing, right? Type in that stuff while they're light adapting. They're ready to go. Same thing. Same thing for red, right? Although you're not going to get as far. So here, what was the intensity the person got to? 16. So you put that there. It took them 317 minutes, seconds to get there. Easy peasy, not that difficult. 3 things with 3 15-minute periods with 3 1 minutes. So it should take you about 50 minutes total to run one subject with a little bit of time to get settled. That's why I said please allow 2 hours, 2.5 hours, 2 hours, 1 hour per person's quarter. Questions? Yes?
So do we type the data onto the Excel sheet on the computer in the lab room, or is there
On the one there, make sure you save it, and the next person will come in and just start the next row, right? So make sure you're saving it because it won't save automatically. Okay, go ahead and close it when you're done. And I will go in there and collect all the data and compile it next week before our stats session. You don't have to upload your data to a group data sheet or something like that. Cool? Pushing about 30 minutes here, Pittman. All right. Okay. Before we get, well, let's do So, what are we measuring? How long it takes you to read the word, right? So we have sort of that longitudinal data, like the time at which it takes to get to each intensity, right? What else are we collecting? What was the minimum intensity? You're going to write that in, right? Minimum intensity you reached. And there was one other thing that you were collecting. That you're typing in. The time to get to minimum intensity. What are our independent variables? Whether or not you're in red light. What else? Color of the word. Cool? All right, everybody understands what we're doing and why we're doing what we're doing. And what your prediction, you could write a hypothesis now if you wanted to write a hypothesis about whether or not the red light's present or not, and about what the time to get or the level of intensity would be for blue, green, and red. Color awesome. Okay. All right. You also this week need to be working on your introduction and methods for your lab report. APA style lab report. Don't look at me with blank faces. You did this at least twice in biopsych, 3 times. And in 160 and 161. Okay. And if you took cognitive or social, you should be right, not your first go around. So what are the things that are important to include in the introduction? Probably a description of the rods and cones and the differences between them. Probably should be a paragraph, right? Short paragraph, but it should be a paragraph, right? What are some other things?
Previous literature.
What's that?
Previous literature.
Previous literature? We're not going to focus on previous literature, but you're right, that goes in the introduction. I'm not going to make you look up an article. Again, I'm just feeling really nice.
What the actual adaptation process is.
Yes, right? So, well, first, you probably need to know, describe what transduction of light is, that you have to activate photopigment, right? Light activates photopigment and uses it up when it's transduced. But then, a description of both light, I'm going to give this to you, you don't have to type this up. I'm going to give you this picture. But probably need, these are probably each a paragraph, right, in your introduction. What is light adaptation? What is dark adaptation? Why does it take time? Okay. What else? What about, yes?
Say the different color wavelengths.
Yes, right? So something about spectral sensitivity of rods and the fact that they're not getting bleached in red light. We need to understand these in our introduction, right? And then lastly, we need a paragraph that's describing what the experiment is and what your hypothesis is. Cool? So that's not bad. That's like 6 paragraphs in the introduction.
So since we're not finding articles, you want us to cite the textbook or the
Yes,
slide?
both. Right, so for this information, you can cite both my slides and the textbook. And I will try to remember to post an example of how to cite the slides, because that's an unusual thing in APA format for you, okay? But yes, you only need to do that stuff, right? Everything you need to know should be found either in the PowerPoint lectures or in the textbook. Still have a minute, but I'm done. So you get 1 minute of my time, okay? All right. Go to it. Have fun. Questions?